Jason. Okay, good.
Okay, can you hear me?
Yes, perfect.
Okay. Thank you very much, Jean-Baptiste, for this kind introduction and reminding me about my old papers. That's very nice, actually. In fact, there will be some connection with one of the things I'm going to tell you about. Okay, so so I would like to tell you about some things I've done in collaboration with many people I'm going to cite later on trying to understand and to model some biological systems using simple unsupervised learning techniques. And I'm going to make this all this more detailed soon. But let's say, so what is our motivation? So our motivation is to model complex systems. So by complex systems, I mean systems which are actually characterized by a large number of internal components. Interaction at all scales, spatial scales, temporal scales, between those components. Heterogeneity in the sense that not all pairs of internal components actually interact in the same way. You may have also time-dependent behavior, a lot of stochasticity in the interactions, in the behavior, and so on. And this is obviously extremely challenging from a first principle approach, which means that the a priori knowledge we have about those systems, what we know is not sufficient to be quantitative in the modeling, usually. And the problem is that those systems are ubiquitous in science, especially in biology. And I want to mention only 3 of them. But of course, the list could be much longer. In particular, I think you have all heard about how to predict structure from sequence for a protein, how this is a hard problem that people actually have tried to tackle for decades. And until recently, and with the use of machine learning, it was very hard to do anything. And I'm going to talk about that a little bit later. And another problem, which is selection and coevolution of the immune systems and pathogens in our body. So when we get infected by a virus, for instance, or bacteria, then our immune system enters into play. And there is a kind of arm race between the evolution of the viral sequences and the one of the immune system. And this is a very interesting coevolution problem. And there is an aspect of it which is related to cancer that I'm going to mention also. And a last problem that I don't think I will have much time to discuss is how you can, from a large neural population activity, try to understand what is the high-level computation that this population is doing, and how you can manipulate this population also through some stimulations. Okay, so there are some common constraints and requirements on this problem is that usually the data we have access to are limited, both in quantity and quality. So what I mean by that is that we may have access only to a part of a system, which is recorded or which is measured experimentally. There might be a limited quantity of data. They might be highly affected by measurement noise and so on. Another aspect also, which is very important, is that performance in the modeling matters, but that's not the only factor that should be taken into account. Interpretability is also something very important. We like the model to shed some light on the mechanisms that are actually controlling the behavior of the biological system. There are also aspects which may be in some cases important, like the generative features of a model. So we would like to be in some cases able to generate new data, possibly with tunable features. And I'm going to mention that in the generation of aptamers in a while. And of course, and then there is a last requirement, which is more which may be not of interest for biologically inspired people directly, but I think is important for people doing theory, which is we like to achieve some kind of reasonable theoretical understanding of the machine, of the model. How does it learn from the data? How does it work once the training phase is over? And I think it's important to have some control about what the model is actually doing. Okay, so to make all these things more concrete, and I want to shift to applications now, and I'm going to talk about the first 2 I've listed here. And one way to unify them is to talk about selection. So selection is obviously a fundamental biological process. It's actually the definition of what a biological system is. This is a system undergoing Darwinian evolution. And selection is all over the place in biology, and I want to mention just 2 sides of the same coin in a way, which is selection in rounds of selection. So what do I mean by that? So for instance, here is a complicated drawing of a process that we all know about, which is how our immune system reacts when we get infected by a pathogen. So in our body, we have some specific, let's say, small systems, which are called germinal centers, which contain naive B cells, which are immune cells. And when we get infected by a pathogen, these germinal centers get into, they start forming and they start developing. And the naive B cells get in contact with some pieces of a pathogen, for instance, some parts of a proteins of a virus, for instance. And what is going on then is that the naive B cells are going to try to bind. They are binding or not, depending on their composition, on their sequence, with the pathogens. And you get a sequence, a cycle of selection that is and reproduction in the sense that the cells, the B cells which bind better, which to the pathogen are going to be amplified, and their numbers are going to increase faster than the other ones. So at the end, after a few rounds of selection like that, you get cells which actually bind quite well to the pathogens. And these cells can be used for the body to initiate some reaction, for instance, to kill the pathogens, or they can be used as memory cells, which can be reactivated months or years later when you get infected again. And this is exactly the principle of vaccination. Another example of this selection process is now not in our bodies, not in vivo, but in vitro. And this is used by many people in biochemistry or in chemistry that want to actually build some molecules with some interesting properties. So, for instance, imagine you would like to build some kind of small DNA or small RNA molecules which is able to bind some target. I will show you some practical example of that very soon. It might be very difficult to design it ab initio from first principle. So what people do, they use what they call a SELEX experiment, which stands for Systematic Evolution of Ligands by Exponential Enrichment. It works in the following way. In a tube, you can build a large number of sequences which are purely random. And then you put them in contact with the target. Imagine it's a protein here of interest. And some of them will be sticky and they will bind to the target, another one will not. Wash away all the bad ones, discard them. You get only the bound one that you can release, amplify them with some molecular biology techniques with an enzyme. So you get more of the good ones and just repeat this process. By doing that, you are selecting the good one, and after a few rounds, you get very good binders. This has a lot of applications in bioengineering and so biomedical application to invent new molecules for therapeutic applications. So typically, when you look at this kind of experiments, what you get is the following. You get data, which is a list of sequences. And here this is just a fake list here that I just wrote. So suppose DNA sequences, for instance, you get the sequences and you get the counts, which is how many sequences you have in your tube. Actually, it's a very sparse sample of your tube that is going through high throughput sequences. And you may have 100 copies of the first one, 1,600 copies of the second one, and so on. And some of the sequences will have very low counts, of course. And you get these count numbers at round 1, round 2, round 3, and so on. And from that, you would like to understand what is going on. And that's not easy. So what people do usually is that they define some proxy for the fitness, that is the goodness of a sequence. And what they do is they simply take the number of counts at some round, let's say R, and they divide by the number of counts of the same sequence at the previous round, C of R minus 1S. And that is called the relative enrichment, which depends, of course, on the sequence and also on the round. And of course, imagine that if this ratio here is larger than 1, then you get amplification. So you get exponential growth. That means it's a good sequence for the particular thing you are testing. If it's lower than 1, it should be bad, right? The problem with that is that, first of all, we don't really understand what is going on by looking at these numbers for many different reasons. The counts may be very unreliable. There might be biases in the subsampling when you take a subsample of your tube, of your reactor, and you get access to a very small number of sequences, typically 10 to the 5, 10 to the 6. There might be a lot of sequencing error along the sequences. So two sequences which appear to be different are actually the same. And there might be any problems, especially if you have also sampling errors. If you have very low counts, do you think they are reliable or not? That's not obvious at all. And also the biggest issue, I think, is that even if you are able to approximate these numbers, then they don't tell you anything about what is going on, because you may find that the relative enrichment of some sequence is 5. but we don't understand where along the nucleotide or amino acids composition content here, where is the feature which is very important for the binding and the biological function, okay? So we don't understand what are the interesting features here. Okay, so in order to model that, and I'm going to show you the kind of technique we use, which are extremely simple, dates back from the 80s. We can use unsupervised learning. So the idea is that if we have a data and we like to learn this distribution and extract some features, then we can use architectures as, for instance, variational autoencoders or restrictive Boltzmann machines, which essentially take the data configuration, build some representations, and can assign some probabilistic core to any data configuration, even data which are, of course, not in the training set. And that can be used if we are interested in, for instance, generating new configurations. So the particular architecture we looked at is a very simple one, which is composed of 2 layers only. We have a first layer here, which is called the visible layer, where we put data configuration, that's the X layer, of size n, depending on the length of the dimension of the data. And we have a second layer, which is the representation layer, where we have the latent variable z's here, which is of different dimension priori. So the model is a graphical model on this bipartite graph with some couplings wik, and it's defined as following. So the log of the joint probability of x and z, which are these 2 high-dimensional configurations, is made of 3 pieces. It's a sum of 3 pieces. The first one is a sum of local potential acting on the configuration components here, which are parameterized by some parameters theta I. Then you get the same thing on the latent layer here with another potential g, which may actually is parameterized by some theta k for unit k. And you have some couplings in between the 2 layers, which are telling you how the latent representation are going to extract some features from the data, and in turn, how you can generate new configuration from the latent configuration. So how is this model trained? Well, if you integrate over the latent variables, you get the marginal distribution over the data, and we can maximize these probabilities here, these likelihoods here, over our data set. And we do that, so we maximize on the different parameters, theta I, theta k, and w in the system, in the model. And as I said, not only the marginal is interesting for learning, but of course, when you are training the model, you can actually sample it. And I'm going to talk more about that. And you can, for instance, extract representation from the data. And if you have done a good job in training the model and using some tricks I will explain maybe briefly later, then you can expect that these latent variables are going to be biologically meaningful. And in turn, if you have interesting latent representation, then you can use them and design some data with interesting features. So why do we focus on RBMs? So as I said, they are all modeled from the end of the 80s. So they were shown to be universal approximators, but that's not a very useful result in the sense that this result is holds if you have an arbitrarily large dimensional representation, so very large hidden layer, which is not the case in practice because we are limited by the number of data available for the training. So what matters most is that they can effectively be trained. And there have been new algorithms in the 2010 by Hinton and collaborators, contrastive divergence, which make them quite efficient to be trained. And the reason why we like them is that they are one of the very simplest model implementing the data representation duality, which means that you don't need so many data to train them. And this is a situation which happens very, very often in biology. We don't have so much data. We'll show you some examples. Still, even with a few data, we can use them as a generative model. And one of the reasons we like them compared to VAE are also very good architecture. But one aspect of VAE, which I find a little bit puzzling, is that the latent variables are assumed to be Gaussian, and this is not something which is related to any features of the data, but only just for convenience. And it takes some layers in order to turn any kind of distribution of data into a Gaussian distribution. And one aspect of the depth of the VAE is actually to do that. So with a single layer and appropriate potential on the latent variables here, we can actually achieve the same performance. And other reasons, and I want to mention them, they are important. I will show you some examples. They are largely interpretable. That means since we have a single layer, we can try to look at the weights and understand what they mean. And also, and this is very important, they are amenable to theoretical analysis so that we can understand and control to some extent what they are doing. Okay, so I will show you here three different works. The first one is how we can actually take advantage of in vitro data, in vitro selection process to design new biomolecules. And the particular example I'm going to focus on is thrombin. So thrombin is an essential protein for the coagulation of blood. So thrombin can actually exist in two different forms. One is thrombin itself, which is soluble, and it can actually bind onto itself and if you have a polymer of thrombin, then you get a fiber, which is at the basis of protein networks, very important for for coagulation, sorry. So if you want to prevent blood from coagulating, then you have to prevent two thrombin to bind onto each other. So one way to do that is to actually engineer some very small molecule which is going to bind onto the binding site of thrombin so that it doesn't get sticky anymore and it cannot bind to another one. So people have been looking for these very small binders, ligand, and they use SILEX, the kind of experiment I've shown you before, to do that. I'm going to tell you how we can actually engineer new ones using RBMs trained on those data. Another kind of selection process in biology, which is actually very complex, it's not in vitro anymore, this is inside our body, and that's related to what is going on in the immune system when people have cancer, in particular pancreatic tumors. So pancreatic cancer is a very bad one with a very low span, life expectancy if you are diagnosed, about 7 months on average after surgery. And but it turns out that some people actually are long-term survivors. They can live for years and the tumor doesn't seem to grow. So why is it so? And there have been some hypotheses about that. And there are a few data, and I will tell you what we've been doing. And the last aspect I would like to mention is much more theoretical. Let's forget about data and try to see how this machine works. So in particular, if you have trained your particular RBM model on a set of data of interest, then you have defined a probability distribution over your set of configuration. This may be very complex with a lot of modes. How can we be sure that actually we are going to explore the whole landscape and we are not getting stuck somewhere here? And I will show you how actually combining different RBMs and building a stack of different RBMs can help to doing that. Okay, so let me start with the first problem. So here is a brief reminder how this SILEX experiment works, and this is some drawing I took from this paper, which was published in 5 years ago. So what these people have been doing, they were interested in finding good binders to thrombin. So they have thrombins here, which are actually exposed, attached to a magnetic bead, and they are in the solution, aptamers, with a core here, which is a scaffold, which is not changing. It's always the same in solution, but they have flexible parts here, the heads here. It's a double head here, double loop, which is flexible and random in the initial library. So just to give you some examples, given the length of these flexible parts here, and given that on each side you may have 4 different nucleotides, there are DNA aptamers, then you have about 10 to the 24 sequences which are in principle possible. In the initial library, which is done randomly, there are about 10 to the 15 sequences which are present. So it's a very small, it's a sparse sampling of the total space. Now what these people do, they take the library, they, as I said, try to see whether they actually bind here on this target here, and they just repeat this process by amplifying the good ones. So essentially, what you can imagine is that there is some kind of landscape which is totally unknown here, which I'm trying to draw. The sequence space is the x-axis, which is huge, and you have a vertical axis which is telling you, well, it's a good binder if it's high, and it's not a binder if it's low. Something like that should exist, right? So at the beginning, at round 0, since we have a library which is purely random, there might be a good ones, some good ones, but most of them are actually very bad. So they are below here, in the bottom here, and few can be actually better. Now, if you increase the number of rounds of selection, you start by having better binders here. And if you go up to round 8, which is the maximum number of rounds that was tried that was tried in this experiment, you get very good binders, excellent binders. And one of them, thrombin AD, if you remember about chemistry, has actually a binding affinity constant, which is extremely low here, 340 femtomolars, which is quite remarkable. Okay, so what we wanted to do is we have access to some sequences which were taken by a subset here of what is inside the pool. And remember, there were about 10 to the 15 sequences. And what people do, they take a small volume inside the pool here, the sequences, and they get about 10 to the 6 sequences. Most of the sequences are actually the same. So that means the unique sequences is about 1,000 or 10,000, because you get the same sequence with multiple counts. From that, can we build a model and try to understand what is this function? And can we engineer new binders? And so we would like to do different things, design evolvable binders, new ones. And we like also to be able to predict selection, which is a kind of dream in selection, right? Even in vitro. So essentially, if we take the data at round 5, can we actually build a model which is able to tell us what is going to happen at the next round? Okay. And I insist on the fact that we take only one round to train the model. We don't look at what is going on between 2 rounds. So can we actually predict the future by looking at a snapshot of what is inside the pool at round 5? We start from round 5 here because we don't have access to the previous rounds. They were not sequenced by the experimentalist. Okay, so what we do is we take our RBM model, we just train it, as I mentioned, through maximum likelihood. And we get, what we can do is we can plot on a test set the histogram of the log likelihood at round 5. And this is trained at round 5. So you get this kind of histogram here. So x-axis is the log likelihood. So good ones are here, bad ones are there. And you get a 3-peak histogram like that. Now, if you take exactly the same model, so we don't train it again, but you just plot the histogram with the data at round 6, round 7, and round 8, these are the histograms you get. So you see that the low peak here is disappearing, and the middle one here is actually shrinking in height, and this one, the most right here, the rightmost peak is actually growing. In fact, it can be seen experimentally that these peaks here correspond to non-binders, and these 2 peaks here correspond to aptamers which bind the thrombin with both loops. They are the very good ones. Only one loop is binding the thrombin, and they are intermediate binders. So we see something where the bad guys are actually disappearing and the good one gets amplified, but does it have anything to do with fitness? Again, the model is trained on a single round. So we go to the usual definition of the relative enrichment here, which is the log of the number of counts of a sequence at round R over the same thing around R-1. And we expect this guy here to be a proxy for fitness. Now, if we plot what people do in experiments in data analysis against the log likelihood of our model, this is what we get. So we get the log likelihood here, the log enrichment at different pairs of rounds. So you have different colors for round 5 to 6, 6 to 7, 7 to 8. And you see that the relationship, except on the on the edges here where we don't have much statistics, is actually pretty much linear. We get almost a linear relationship. We think that the log likelihood of the model is actually a good proxy for the fitness, the goodness of the sequences. And the slope here of this linear relationship is decreasing with the number of rounds. That is not very surprising because actually people don't repeat always exactly the same experiment. They change the intensity of washing. So it means that the intensity of a selection can change from round to round. So if we take just the exponential of that, you see the number of counts at round R for a sequence here. If you just take the exponential, it's given like this. And if you just iterate it from round to round, you get that essentially what you are doing is that you are looking, you have up to some factor here, which depends on your initial condition. You are looking at what is going on with your model, predicted to the power here, which is actually an expression of a cumulative selection strength at the different rounds. Okay, so we can now try to interpret what the machine is doing. And so we can look at the different weights of the machine, okay, of the machine corresponding to the different representation units here, the different latent variables. So I'm just showing you different weights. So here on the x-axis, you have the position along the aptamer. You have 40 nucleotides. And here on the y-axis, you have some letters. So these letters is kind of a local representation of the intensity of the weight. So that means, so K here is the hidden unit, which is fixed. So it's K1, 2, or 3 here. And I is this position along the sequence of the aptamer. And we just present the intensity of W. If it's positive, it's going to be above the 0 line here. If it's negative, below. And since we have 4 possible nucleotides on each side here, then you have a letter corresponding to the different nucleotides, and the height of the letter is proportional to W. Okay, so if we look, before we look at the weight, we look at the average profile, which is just the histogram of the frequencies of the different nucleotides on each position. We see some motifs here, which is called a G quadruplex motif that biochemists knew about, because they have done exactly that. Here, we don't need any model to do that. It's just statistical data. But now, if you look at what the machine is capturing, we see, for instance, the first thing here is concentrating on the second loop. And if you look at the alignment of the GGGG, it's just exactly the GGGG here. So it's one part of this quadruplex here, which is shifted by 1 unit. That means that some of the aptamers are actually a little bit more flexible. They are lengths. There is one more nucleotide in between these 2 parts of the quadruplex here, and everything is shifted here to the right. So you see a kind of a variety here inside the loops. We see also a correlated shift of the boundary of the GG duplex on this motif here. And we see, for instance, a motif that we don't have any idea about what it's meaning from biological point of view, and biochemists didn't know either, which is a long-range AT rich motif here. So it's interesting because the machine is capturing something which we should try to understand better. Okay, so it's really able to unveil some new biochemical motifs. So now what you can do is, once you have a machine, you can actually try to use it to design new aptamers. And we have done that. And we have tried to design things which were interesting. So for instance, we try to design new binders, and these are the yellow points, which are predicted to be very good by the model, but which were not present in the data. Okay? Probably because they were not actually initially in the library, so they were never detected, they were not there. But the model said that they should be very good. We can also look at some of the binders or aptamers which are in the data set, but they have very low counts. But they are predicted to be good. And we don't understand why they have low counts. But I told you that there are many reasons why actually count is very unreliable measure of the goodness of a sequence. And we can also look at very good binders, which should become very bad after 1 mutation. And so we tried different things here, and we did different predictions, and we had a very high accuracy compared to the results of experiments. So this is an example here that actually training a model with few data on selection experiments can lead to interpretable features in the data, can unveil interpretable features, and can be useful for design. So we have done much more on design. I don't have time to explain everything, but I just want to say that we actually use similar models on proteins to design, for instance, new enzymes, which are very important for the metabolism of bacteria. So what we do, we train the model exactly as I told you from some data. So they are not in vitro selection data here. They are just data sequences coming from the same enzyme in very different bacteria, very different organisms. We can, once we have learned the model, we can sample it using Monte Carlo methods and generate new sequences. You can synthesize the corresponding genes, insert them into bacteria where the native gene has been deleted, and you see that this inserted gene is actually able to rescue the wild-type gene, and you have a perfect bacterial growth. And we have published that a few years ago, and we have done that also for other proteins, and it worked really well. An interesting aspect of that also is that we can actually, if we believe in the model, count how many non-natural version of this enzyme exist. So nature, in the course of the last billion of years, has actually sampled some of those possible proteins, but there are many more which have never been explored, and the model is giving us some idea of the number of possible proteins. And we get, for instance, for corresponding mutation, something like 10 to the 25. We have also done application on enzymatic RNAs, which are important in regulation. And I want to mention briefly also mutational paths, where you can take, for instance, where we try to design not only a single protein, but actually a path of proteins. So you can start from a protein which is binding different ligands with different classes of different ligands from a chemical point of view. And so you take one example, which is binding here some kind of ligand, another example here, and then you're trying to find intermediate proteins and see how the function is actually changing along the path. And one application is what we are trying to do is to do ancestral reconstruction, because it's known, I mean, it's believed that ancestor proteins were more promiscuous than modern proteins. So they may actually have been less specific than the proteins we know of now. And we try to see in between if we can find some proteins which are less good at binding both ligands, but they can do, I mean, each ligand separately, but they can do a little bit of both. And this is what we see actually, and this was checked by Marco Ribezzi and Hamed Rian at ESPCI recently. Okay, so I want to go now to the second application, which is going to be fast, but it's another problem related to selection where the data are even worse than the data I mentioned. So here we are interested in this aspect I already mentioned very briefly. So the question is whether actually we can understand why some patients are long-term survivors of pancreatic cancer while other ones are not. Most, unfortunately, are not. And one of the hypotheses that was put forward by researchers in the field is that in long-term survivors, it might be that the immune system is able to recognize the mutation associated to the tumor, and the T cells, which are one part of our immune system, is actually targeting these mutations. And it's always keeping the tumor under control. So the tumor will evolve, new mutation will appear, but then the T cells of the patient are going to adapt and always recognize this and maintain the tumor under control. Okay, so essentially the data we had access to, thanks to a group in New York, in Memorial Sloan Kettering Cancer Center, were data about 30 patients, where there were a few pancreatic tumor long-term survivors and more short-term survivors. And for each one of these patients, due to surgery, we had access to the tumor mutations. And we had also access to a sample of T-cell receptor sequences, which were obtained by actually sampling the blood of the patient. And one of the questions we were asking is whether any kind of evidence for the fact that the T-cell repertoire was actually linked to the tumor mutation. So was the population of T-cell tumor dependent, neoantigen dependent, and whether any relation between this and the prognosis. So it's pretty tough actually to do any kind of thing like that, because essentially the data are very poor. So what also we had access to in terms of experiments is that the T cells, which were actually sequenced and obtained from the different patients, could actually be tested against the tumor, parts of the tumors here with the right mutation, the one found also in the same patient. And people could actually measure the evolution of the number of T cells in a process which is very similar to the one I explained just at the beginning as a function of time. So you would expect that if the immune system has recognized the tumor mutation, then the T cells are adapted to this tumor. So that means if you put them into the presence of this mutation, then the numbers is going to grow very fast, specifically in response to that. While if you present another part, for instance, of a wild type non-tumor tissue, then you will get no expansion or much weaker expansion. Okay, so we have access to this kind of data. So essentially what you get are the sequences of a T cell here. You get some counts. This is the typical number of sequences we get with the number of counts here, for instance, one of the patients. And you have the data at day 0 and at day 21, where these T cells are grown into the presence of the tumor mutation. So what we do is that, again, we can use our machine here. So we train our machine with this kind of data, and we define the likelihood. We've had access to likelihood according to this model, and we can compute the difference in log likelihood, the log-odd ratio between the data, the sequences at day 21 after the growth, the expansion, and at day 0 originally. And we can do that with when the T cells are in presence of the tumor mutation, or where they are just in presence of the wild type sequence, which is the control. So the claim we have is that actually the log likelihood of the model is a much more reliable estimator of expansion than counts. And the reason is exactly something I just mentioned right at the beginning of the talk. if you just look at the counts of each sequence, then you are not using any kind of information, cross-information between different sequences. You compare the number of counts at day 0, day 21, and you do that sequence per sequence. What you learn from one sequence is not helping you for another one. So you cannot reduce the noise by having more sequences. While the machine is trained on all sequences together, and in fact, it's going to give you a consolidated score, which incorporates all the information. And using that, we could, for instance, obtain this result. And I just want you to look only at the x-axis here. So the y-axis is something else I'm not going to discuss. So here you see points. Each point is a patient. And you have in red the short-term survivors, and you have in blue the long-term survivors. And what we measure here, we take our machine and our model, and we select the top sequences with according to the log likelihood of the machine, which are not, again, the sequences with the most counts, okay? And these are sequences of T cells. And we look at them, and we look at how much they are similar or not. And that's what we call the dissimilarity index here, arbitrary units, doesn't matter. So what we see is a clear separation, which is statistically significant, between low dissimilarity here for long-term survivors and high dissimilarity for short-term survivors. So it seems it's a kind of clear indication that actually in long-term survivors, the T cells are really focused on the specific mutation. And this is why they are similar, because they all look at the same kind of mutation, so they are close to each other in terms of sequence and properties in order to adapt to this mutation. While in short-term survival, they don't have access for some reason to the mutation, they don't focus on it, so you get a much more diverse set of T cells, which is unable to detect and attack the tumor. So following this work, actually, this work gave some hope, because it means that actually, if you were able to actually have the immune system focus on the tumor, then maybe something good could happen. So maybe you have focused, you have seen this paper, which was published last year in Nature, or maybe you have seen it in journals. So for instance, this is just something I extracted from New York Times last May, but I think there were also some account in Le Monde, where actually the people were our collaborators here who are the origin of this study and who are in New York actually used exactly this approach in order to build a new vaccine. So what they do, and this is summarized here in the bottom plot, they take actually after surgery a small piece of the tumor. They can sequence it to try to find the mutations. And they have also modeled for the TCR sequences, the T-cell sequences I mentioned to you. And they're trying to find, given the repertoire of the patient, what could be the best mutation more likely to be recognizable by the immune repertoire of the patient. Now, once they have found it, and that's based on a computational model, which can be criticized, but I mean, you have to do that with very few data. Then what they do, they just synthesize RNA of this part of the tumor, and they use exactly the same technique that was used in order to do the vaccine against COVID, right? mRNA vaccines. So they use these RNA vaccines, and they actually work with the BioNTech company, the one which was working with Pfizer 3 years ago. And you can actually use that as vaccine for a personalized vaccine for people. And you just want to boost their immune system to recognize this mutation. And of course, that was a very small sample of people, but actually they had very good results on 8 of 16 patients, which is, so it's 50%, and it's a huge number for this cancer, which is actually very, very tough. So there is a lot of hope that these techniques, if they are improved, can actually be very successful for immunotherapy for this very nasty cancer. Okay, so I want to move to the last thing I'd like to talk about, so I will brief. But then I would like to move away from data and go more into theory. So suppose you have learned a machine from your data, and I already gave you 2 examples, and now you would like to know whether this machine is going to be easy to sample. Can you actually explore the landscape, probability landscape defined by the machine? So usually, the problem is the well-known problem in many fields, in applied math, in statistical physics. We have a distribution of probability P of X. and we like to sample it. And usually what people do is that they use Monte Carlo Markov chain, which is most of the time local in the space of configuration, but there are plenty of variants here. And you try to have a dynamics which is guaranteed if you wait long enough to sample from P of X. So how does it work for RBMs? For RBMs, that's the algorithm that people use is called alternative gate sampling. And it's very easy. It exploits the bipartite nature of the interaction graph. You start from a data point. You just sample from this data point one representation, latent configuration, and then you sample back and so on. And why is it good to do that? It's simply because actually it's very easy to do. Because condition on the data configuration, all the hidden units here, all the latent units here are independent. So that it's extremely easy to sample from them. And then you do that exactly in the same way, and they are independent again in the other way, and you just repeat this process. This is guaranteed to converge, except that if your landscape is quite tough, then actually what happens is not very nice in most applications. You may be in a situation where you start from a data point, you go to some representation, you go to another data point, another representation, and then you go back to this. Essentially, you get stuck in some kind of local maximum of a joint probability distribution, and it's very difficult to explore other parts here of the data space and the representation space. So there was some ideas in the machine learning literature that actually going to deeper machines could be better. And I'm citing just two of these papers about 10 years ago. And the idea is that when you go deeper in architectures, then representation may be more coarse-grained, and the corresponding landscape are simpler to sample. They essentially extract invariants from the data, right? More abstract representations, which actually forget or can omit all the details in your particular data configuration and extract some kind of general feature. So if you get some kind of simpler landscape here after a few layers, then it would be very nice to try to exploit this idea in order to sample the bottom layer here, which is the one we are interested in here. So we use this idea and propose a very simple application of this idea, which is quite effective and very simple to implement. So essentially the idea is the following. So here, the bottom machine here is the machine, sorry, it's the machine I've been talking about over the last half an hour, 45 minutes. So here is your data configuration. Here is your latent configuration. Okay, so you have learned this machine according to your maximal likelihood algorithm. And now what you can do is actually use the representation as some kind of new data for another machine. And the only requirement is that the other RBM have an input layer here of the same dimension as the representation layer of the bottom one. And then, of course, you can have another dimension, usually smaller, for the representation of a second machine. And you see, you can actually iterate this process and have a stack of different RBMs on top of each other. So the only machine we are really interested in here is the bottom one. But all the other guys are going to help us a lot to sample the bottom machine. And the intuition for that is that we may have actually clusters of data in the configuration space that correspond to different representation. And if we sample back and forth, then we go to this representation and we sample back always the same cluster. Very difficult to hop onto another cluster. But if we have another machine higher here in this hierarchy, then maybe all these representations are going to be mapped onto the same one. They are part of the same cluster for this machine. And then we can move back and explore very efficiently other configurations which are very far away. So this is the idea. And essentially what we are doing to do is that we are going to run. So we start from our data, train the first machine, use the representation as metadata for the new machine and so on. And then we just run independent Monte Carlo Markov chain for all these machines, back and forth algorithms. And from time to time, we try to exchange the latent representation here with the data configuration of that one through a replica exchange. And of course, you have to do that maintaining detailed balance, which is you just have to check that the difference somewhere in the log probabilities here is actually satisfying detailed balance. It's easy, and it doesn't require to compute the normalization factor of probabilities as in usual Monte Carlo. So it's computationally efficient. Okay, so as an example, we have done that for instance, MNIST. It's a toy example. We take MNIST 01, so we take only 0 digits and 1 digits. They are very dissimilar. So if you train an MNIST RBM only on 0 and 1s, It's very tough. So you can generate very nice zeros if you start from a 0, very nice 1 if you start from a 1. But it's extremely rare that you can jump from a 0 to 1 because they correspond to two different modes in the distribution of the data, which are very far away from each other. So we used our technique here with a stack of 4 different RBMs. And here are the results we get. So if you take only one RBM, you start from the 1, you get stuck. Here are different snapshots at different times on the input layer. And now if you take 4 RBM plus these techniques of exchange, then you get a nice exchange. Here you can go from 0 to 1 and so on. And here you see the number of class exchanging as a function of time, which increases very quickly with our techniques, while it's 0 essentially with the usual back and forth. And our technique is actually even doing better than parallel tampering, which is called also replica exchange Monte Carlo, which is a usual technique that people use, where essentially they divide a chain of model. You want to sample from P of X, it's very hard. And so you will sample from other models with P of X to some power, which is between 0 and 1. So when the exponent is very low, so this is a very flat landscape, so it's very easy to sample from. And then you create a chain with exponent growing up to 1, and you try to exchange along the chain here, so that the fast exploration is going to actually benefit here this model, the one you are interested to sample from. So one of the questions we ask is why this works. And can we understand for which parameters this works? And this is what we looked at, and I want to be very quick on that. So because I don't have much time. So we can actually deal with this analytically with this model when in the limit of over-parameterized machines, where the size of the input layer and the size of the representation layer goes to infinity at a fixed number of data points. But in order to make the number to make this model non-trivial, we impose some strong regularization on the weights. And we can use statistical mechanic techniques, which are not rigorous, but we believe give exact results, like the replica method from the statistical physics of this order system, to understand what is going on. And essentially, I'm going just to keep going. We can compute the mixing time of one machine. We can compute the mixing time of the second machine using the representation as data for the next one. And we can compute also the swapping time between this hidden and visible configuration here. And essentially, if the mixing time of second machine, the maximum between and the swapping time is smaller than the mixing time of first, it means it's actually going faster. It's going to be very useful to use that because this machine is very fast. The swapping time is also small. So it's better to go up this way and go down than actually mixing only from that one. And that gives condition on regularization aspect ratio, which can be actually checked in real data, and that helps train our machine. Okay, so I will stop here. I try to convince you that restricted Boltzmann machine, all machines from the 80s, they are simple and versatile tools for generative and supervised learning. They are both interpretable and amenable to theoretical analysis. There are many other direction applications I didn't have time to mention. For instance, how to disentangle representation using annotated data to try to align some direction of a latent space along some biologically interpretable features. We have a paper on that. And I think it's an exciting field at the crossroad of biology, machine learning, and statistics. And that's what I'm interested in. So I want to mention my collaborators. So in particular, Simona Coco at ENS in Paris. Many other people, in particular, also Jorge Fernandez de Custodias, who is a postdoc in the group. Jérôme Tubener, former PhD student, now in Tel Aviv. And also many collaborators that I'm citing here. Okay, thank you.
Thank you so much, Remi. That's wonderful. And are there already questions, or should I start? You can open your mic directly and ask the question. I maybe don't want to start. So the nice part about what you talked about is the tractability of the models that you're able to generate. What are your views on the fact that now there are many projects starting on the biological sequences involving LLMs, which are extraordinarily efficient at capturing very large-scale correlation, but are large black boxes, or just at the moment? How do you feel that this type of the interplay between the two for modeling and getting results?
Well, you can see it in two ways. You can see it as a very strong incentive to try to understand what LLM is doing. I think it's a very nice way of seeing it, right? So it's a very strong push. So theoreticians should push a lot in this direction. Otherwise, it depends on what you want to do. I mean, if you want to do bioengineering, maybe in some application, you don't care so much about understanding what is going on, except that since you are not understanding, then maybe you are missing nice, very nice things, and you could do actually much better by understanding what is going on. But of course, I mean, maybe it's my bias as a physicist. I would like to have some control about what these machines are doing. But I think we are trying also to push this simple machine in a more, in deeper architectures to try to use also this kind of ideas. So I'm trying to start from the simplest one and trying to make them more complicated. But of course, I'm not against LLM. That's not the point. But as I try to say also to advocate the fact that in many biological applications, the data are actually pretty scarce. The cancer data, for instance, there are not a lot of data, especially because that should be personalized. So it's one patient. So we need to be careful here.
This is also my next question was about that. And as you mentioned, with your model, there's also a question of mutational path that can be non-trivial, or at least you won't be able to pass from one state that would be favorable to another one due to the complexity. How does your model, I mean, do you don't care because you're able to sample and you will see this other stable point or something like this? Or how are you able to include when clearly going from, I don't know what sequence or one configuration would require multiple steps to get to another one?
So when we sample the path, we don't do one sequence at a time and try to see whether it reaches the second sequence, the arrival sequence. What we do is we use a technique that people have been using in computational chemistry and physics for a long time. You use some kind of strings. Okay, so you don't simulate a single configuration, but you simulate a string of configuration. So if your path has, I don't know, I'm just saying a random number, 50 intermediates, I mean, you run simulation over 50 configurations at a time, and it's anchored on the initial point, anchored at the final point, and then in between it should adapt. And you essentially, you want to have a compromise between good probabilities of the intermediates and also closeness between the intermediates, because the risk is that you will have half of them on the first sequence, half of them on the last one, because we know they are good and nothing in between on a huge jump. That's not the path, right? Okay. So we just simulate the whole thing.
Okay. Questions? I can go on. Then I'll go on. In the last part of the talk, you mentioned rapidly the system you used, mentioning replica. Is it the same replica as the usual replica in when you're modeling phase transition?
Yes.
Quickly. Okay.
Yeah, I didn't want to be technical because that was already too long. Yes, and so that's interesting because so here the problem when you have the probability distribution, the likelihood of the model, essentially, you have so maybe can I share again maybe?
Yeah. Normally you should be able to. Okay.
Yes. So maybe it's good to share again. So let me.
It's a bit quick, so I remember seeing rapidly replica, but.
So let me just show the formula here. Okay. Okay, so if you look at the joint distribution P of xz or the marginal, you can imagine the marginal when you integrate the z. That is, I mean, the normalization constant is quite tough. It's very difficult. Actually, it's intractable. It's actually easy to compute this, the log of P of xz, up to an additional constant, which is the normalization. Okay, if you do Monte Carlo, the only you don't care about this normalization thing, so numerically it's not a problem. If you want to actually do theory, you need to be able to access that. So the normalization is essentially, you take the exponential of these guys, so the sum of the three terms, and you should be able to integrate over all x's, all z's. And so that's something which is not tractable. So what we do is actually we use some replica approach because it's at the denominator in the normalization to put it at the numerator. So with a negative number of copies of that. So we see that 1 over something is the something to the power minus 1. And then the something is to the power n. And then we can do some calculation and then send n, which is a positive integer, to minus 1 at the end of calculation. It's actually worse than that because we are not sampling from this distribution, but this distribution to the beta, where beta is large, to do a maximum likelihood. So we have a large negative number of copies. So there is a process of analytical continuation, which is well known from people doing replica approaches, that has been studied also a lot in the math literature. So far, justifying the replica approach has resisted a lot of works. What has been done, and there are famous papers by Talagrand, co-workers, and so on, and other people, on justifying the results obtained with the replica approach using probabilistic frameworks. And this is one of the, I think, most important results was the proof that Parisi's solution of the Shannon-Kilpatrick model that he had in the 80s was correct in 2000. Okay? So, but nobody is able to justify, to my knowledge at least, the replica approach by itself. But yet, we believe that the results in this particular setting are exact. When we have very large machines and the number of data of the other one, which is a very specific, limited setting, but yet it's possible to do some calculation in that setting. Yes.
Other questions than mine? And with that, thank you very much, Remi, for this wonderful talk.
Thanks a lot.
Thank you all for coming, and see you at the next PRAIRIE Colloquium. Bye.
Bye-bye.